Let's move on to the next session for today's workshop. And this session is going to be an overflow or overview of single-cell RNA sequencing technology. And we are going to get introduced to cell barcoding, UMIs, typical experimental or overview of experimental design. And we are also going to get introduced to typical analysis pipeline, where you will pre-process quality control and normalize the data, and you can apply dimensionality reduction and further clustering of cells and differential gene expression analysis performed for the different cells and different types of cells will help us understand the biology behind the relevant questions and relevant samples. So without further ado, let's get started and we go on. So this flows as a part of with the foundation that you already have some introduction to bulk RNA-Seq analysis. So if you do not have such introduction, so it is very extremely worthwhile to check out the transcriptomic course from OmicsLogic's platform. And once you are through with some sort of basic with transcriptomics course, which deals with bulk RNA-Seq analysis methods and understanding the logic behind them, then you can easily get yourself promoted to single-cell RNA sequencing and single-cell RNA-Seq analysis method, single-cell transcriptomics, because most of the information that we are going to handle today and most of the discussion that we are going to have today and in general in single-cell RNAs or single-cell transcriptomics case will be in comparison with bulk cell RNA transcriptomics only. So we learned that in, or we already know from bulk cell transcriptomics that raw reads can be subjected to a sequence of analysis methods, and then we can obtain a table of expression, and then we can use several visualization methods to visualize this table of expression and perform statistical analysis on this table of expression, and you can also apply differential gene expression analysis on this table of expression and etc. So we are going to find out what are the difference that we are going to get when we are subjecting the reads or subjecting the raw reads into a single-cell RNA sequencing or single-cell RNA-Seq pipeline. So let's, so in comparison with bulk cell RNA-Seq, actually bulk cell RNA-Seq will actually allow us to measure the abundance of messenger RNAs as a population average of all the cells in the sample. Right? This has certain advantages, but this also prevents us from differentiating between changes in regulation within the cells, and also it also helps prevents us from understanding the composition or heterogeneity within the sample of cells. So we will also lose, we lose cell differentiation information, we lose cell heterogeneity information, and we do not also understand or we do not also consider eliminating some of the cells for their expression data being an outlier compared to the other bulk cell transcriptomics pattern. But in single cell transcriptomics analysis, it actually helps us to profile all the messenger RNAs present in one cell or present in single cell by cell basis. And this constitutes the most widely used profiling technology that we now know as scRNA-Seq technology. So unlike unlike its cousin bulk RNA-Seq profiling, the sequencing libraries are generated from thousands and thousands of cells in bulk RNA-Seq profiling. The single cell RNA-Seq technologies isolate each and every cell within these sequencing libraries and then generate a cell-specific sequencing libraries or cell-specific RNA content with cell-specific molecular barcode. So the approaches of attaching a barcode or attaching identifier codes to each and every sample in our library will actually help us in counting or quantifying the expression pattern very efficiently and without subjecting it to any biases that we will discuss in upcoming slides. So for both the approaches in general, as we just know, will generate gene expression estimates at the cell level. But for the single cell technology, it actually enables us to measure the transcriptomic information of thousands and also sometimes millions of cells in a cell by cell basis environment. So, so what does this measurement will enable us to understand, or will enable us to study? So this access to transcriptomics information from the cells on a single cell basis will actually help us to understand the profiles of these individual cells and to learn about the cellular heterogeneity. It will also help us discover new cell population or novel tissue types, and it will also help us order or arrange cells within a developmental trajectory to understand the different differentiation pathways and functionalities that govern these developmental trajectory of these cells. So to again sum up the difference between bulk cell and single cell in a very short summary. So bulk cell RNA, on a technical advancement, it replaced microarrays, and we know that it has been widely used to measure average expression levels of each gene across a large population of cells in a sample. So when you have a homogeneous sample, so performing a bulk cell RNA-seq transcriptomics analysis to understand the expression patterns of these samples gives you meaningful results and meaningful understanding behind the biology, governing the biology of these samples. But on the other hand, when you have single cell RNA-seq, it actually measures the distribution of expression level for each gene across the population of cells. So this measurement of gene expression across the different cell types or different individual cell resolution will actually allow us to study new biological questions which are cell specific. For example, as we already saw, we can study heterogeneity and we can study differentiation, and we can also study the stochasticity or the randomness of these gene expression across the different cellular population. And we can also study the inference of gene regulatory networks across the cells for any given condition. For example, and we also see that bulk RNA-seq studies often do not remove outlier cells. So if you have a cell that does not belong to your bulk of the cell or majority of the samples, then we have no way to remove the expression values from this specific, or expression quantification from this specific cell. But on the other hand, single cell RNA-seq analysis in itself offer ways to remove failed libraries, and based on the percent of reads mapped, based on the percentage of the mitochondrial genome present, and based on presence of the different marker genes, which we will discuss in detail in the upcoming slides. So these advantages, and together with the fact that we will get resolution of expression pattern for each and every cell, makes the single cell transcriptomics analysis a very enticing research field. So some of the major applications of these research field, which we already touched on, but will give you, for example, it can help us understand new biological questions like cell-specific changes in transcriptome, like cell type identification, or cell differentiation along the timeline, or some random expression patterns of the gene and their distribution of expression patterns across the different types of cells that we are trying to understand and study. So these will form extremely important part in understanding the tissue types and how they come together in forming cell types and how they come together in forming specific tissue types in general. So if I can summarize on the whole of a generic single cell transcriptomics pipeline or analysis and quantification, then there are some extremely important steps involved, and just listing out some of the very important steps, we will go through these steps one by one. So first we have to extract the cells from the tissue, obviously, and then from this tissue we have to isolate the cells as a single cell entity. And this is one of the extensive steps which can be executed by involving different technologies, right? And we will discuss an overview of these multiple different technologies which offer us to isolate single cell from this tissue samples. So some steps follow a typical RNA-Seq pipeline that shares its analysis sequences with bulk RNA-Seq analysis, like library preparation, amplification of these mRNAs or cDNAs, and then sequencing in general, and then generation of count metrics, and then followed by downstream analysis. So depending upon the complexity of the data that we have, different downstream analysis can be applied to identify different cell types in the sample. So we will apply predominantly use downstream analysis like Surette analysis to identify novel biomarkers or novel markers for different cell types. And we can also subject these and the output of this single cell RNA-Seq pipeline to construct cell trajectories using differentiation analysis from monocle pipeline or monocle tool, and and likewise other tools. So if you see, or if we consider single cell transcriptomics from a technological standpoint, so single cell has been around since 2009, and so it's a fairly recent technology. But given the amount of cells that can be explored from the beginning of 2009 until recently, we can note an exponential increase in the scaling of single cell RNA-Seq in the past decade. So the technology is fast improving, and the ability to isolate cells on a bulk level, right, has developed enormously. And this has given rise to a new cousin or a new analysis method, which is spatial single cell transcriptomics, which is not been which is a very important single cell technology and single cell analysis field, research field that will help us understand the tissue heterogeneity and cell differentiation types. So, for example, this is an excellent review or overview that is published in around 2017, and this actually contains relevant publications, and it compiles an interesting list of protocols that can be automated or that can be employed to perform single cell RNA-Seq analysis or single cell RNA-Seq data collection in general. So when we talk about data preparation and single cell RNA transcriptomics being different from bulk cell RNA transcriptomics, and major part of that difference actually comes in the preparation of data. So how you can effectively separate the cells for sequencing. So there are methods that can be categorized into different ways, but two most important aspects are the approaches to quantification and the approaches to capturing cells and isolating cells as a single cell from the tissues. So for quantification, we have two approaches or two types again. We can quantify based on full length of the reads, and we can also quantify based on the tags, right? The quantification based on the full read length will actually tries to achieve a uniform read coverage for each and every transcript. But in contrast, when we use tag-based protocols, we only capture either the 5' end, or we only capture either the 3' end of each RNA. So the choice of this quantification method has extremely important implications in what types of analysis that the data can be used for. In principle, a full-length protocol should provide an even coverage of transcripts, and we will see, or we shall see, that there are often biases in these coverages. One of the main advantages of the tag-based protocol is that they can be combined with molecular identifiers or unique molecular identifiers, which can help us in avoiding these biases that arises from this coverage and arises from general amplification procedures, right? And this will help us in improving the quantification accuracies. On the other hand, being restricted to one end of the transcript may actually reduce effectively the mappability, and also it might make it a little bit harder to distinguish between different isoforms of the genes. So this generally the strategy that is used for capture determines the throughput, and also it determines what kind of additional information besides sequencing that can be obtained. So while choosing or opting for the technology that quantifies your reads or quantifies your mRNAs, you have to make sure that you have the kind of analysis also in your mind. As we see, since UMI-based or tag-based technologies may find it difficult to distinguish specific isoforms. So if you have a sample that in which you are expecting different splice variants or different isoforms of your cell, of your genes, then it is better, or it is better served if you can make use of the full-length protocols instead of the UMI-based protocols or tag-based approaches. So there are three most widely used options for isolating these cells into single cells, and they are microwell-based approaches and microfluidic-based approaches and droplet-based approaches. So in the first microwell-based approaches, cells are actually isolated using a pipette or using a laser capture, and then placed into these fluidic wells. So these are microfluidic wells, and one of the major advantage of these generic well-based methods is that they can be combined with fluorescent activated cells, and making it possible to select cells based on surface markers. So that will in turn help us avoid heterogeneity in the cells, or help us avoid selecting cells of specific types for which we want to perform this analysis. And this strategy is extremely useful for situation where, as I mentioned, one want to isolate a specific subset of cells for sequencing. Another advantage is that one can take pictures of these cells also, which can be used to, which can provide an additional modality and useful applications to quantify the cells and to tag or to identify damaged cells or identify doublets in a specific microwell and etc. So some of the drawbacks of this method is that they are often low throughput, and the amount of work required per cell may be really considerable, and it is not worth doing such an analysis for a large number of samples. So if you have a very few samples, and if you want to perform these analysis, that can be extremely useful. But when you have hundreds and hundreds of cells in your tissues or in your samples, then it might not be worth performing this analysis using manual labor or using any other form. So on the other hand, microfluidic platforms such as Fluidics C1 will actually provide an integrated system for capturing the cells and for carrying out reactions that is necessary for all the library preparations that you will need to perform sequencing. So since they provide these provide an integrated system for this for the selection of cells and also for carrying out reaction. They are often high throughput or higher throughput compared to the microwell based platform. So typically only around 10% of the cells that you give as an input or that you take from the sample are actually captured in microfluidic platform, and hence they are not extremely useful and not very appropriate when we are dealing with rare cell types, or when we are dealing with cells with very small number of inputs or tissue types that is extremely heterogeneous in nature. But moreover, generally from a financial point of view, the chips that are used are relatively expensive, but since the reactions that actually help us prepare this library can be carried out in much smaller volume, finance can be made up and money can be saved on the reagents end. So one of the most popular method to isolate these cells into single cells is the droplet-based methods, right? And this method will actually encapsulate each and individual cell inside a nanoliter droplet that is present together with the bead. The bead is actually loaded with the enzymes, and these enzymes will actually construct the library. So in particular, each bead will, in addition to this enzyme, will contain a unique barcode, which will be attached to all the reads originating from that specific cell. Thus, all of these droplets can be pooled and sequenced together, and the reads can subsequently be categorized and assigned to the cells of origin based on these specific barcodes that we put in to each and every one of these beads. So these droplet-based platforms typically have the highest throughput compared to the other two methods, and since the library preparation is carried out at the nanoliter level, and the costs are extremely low, and reagent costs are extremely low, and that compensates for the availability of the system that requires more and more financial investment. So when we use these microfluidics based method, they are because of their high throughput output, and because of the ability to automate the systems, we can apply these in large scale for single cell RNA-seq transcriptomics methods. And there are 5 basic steps before the analysis commences to obtain or to carry out these microfluidics based methods. And they are: first, we have to suspension of cells from a complex tissue to isolate cells. So this is carried out in the droplet based method, as we just discussed. And then we will synthesize primers using barcoded primer bead that attaches itself to different cell types. And then the microfluidic device will then capture cells in the oil droplets with the barcoded beads inside. And once the library is prepared, then the cells are lysed, and so not once the library is prepared. Once the droplets captures the cells, then the cells are lysed, and their RNA is actually hybridized with these beads. And this actually generates a single cell transcriptomes attached to the microparticles that we put in these droplets, and they are called stamps. And these stamps are then amplified in the PCR, and once these stamps are amplified, then we subject that to sequencing. And the sequencing analysis or sequencing actually produces read counts and UMI counts. So depending upon the kind of analysis or the kind of library that we prepare. And these are the counts that these are the table or structured data that we use for further downstream analysis methods.